When you are thinking about how the world works, how often do you think about the tiny forces of nature?
Just about never. What do you mean?
I mean microbes. They play an important role in ecosystem processes, such as decomposition and nutrient cycling.
But what do we know about how these microbes live in nature? It turns out they live in communities just like we do.
In this episode, we talk with Dr. Jennifer Martiny from UC Irvine about her work with microbial communities.
This is Radiobio.
Don't know much biology.
Hello and welcome to Radiobio. I'm your host, Sinenta Pertiwi.
And I'm Lily Pennington. Today we have Dr. Martiny with us from UC Irvine. Welcome, Dr. Martiny, and thanks for joining us today.
Thank you for having me.
To start off, can you tell us about yourself and what you study?
Yes, I'm a community ecologist, and I study the community or collection of organisms in the environment, and my specific research focuses on microorganisms.
So how did you get into studying microorganisms?
Yeah, well, I actually started by studying larger organisms. I did my PhD working on conservation biology, especially of birds and insects, and studying their patterns across the globe, how they vary, why is there more diversity in the tropics versus the poles. And I realized somewhere along the way that nobody had really thought about those questions very much for microorganisms. So that's what drew me into microbes.
That's really cool. So when you say microbial diversity, what do you mean by that? What is microbial diversity?
Usually people think of it in terms of the genetic variation, but you can also think about diversity in terms of what the function is of those microbes, what types of metabolism they have, and what they're doing in their environment. So broadly speaking, there's diversity in microbes, in different types of microbes. So fungi or bacteria, viruses. And so you can think about that whole collection of those microbes and how much variation there is among the organisms.
And how is microbial diversity essential for how the world works?
Well, it's quite essential. Even though we can't see them, they're a part of every environment we look at. In fact, even just trying to get them out of the environment is pretty hard. You know, there are people that focus on exactly that. So there are people that actually work on trying to sterilize different types of satellites or ships that might end up eventually going to Mars. We don't want to contaminate Mars with life on Earth. And so their focus is just how to remove microbes, and it's remarkably difficult. So everywhere we look on Earth, we see them. They're in our soil, they're in our water. They're also all over our bodies, inside our bodies, and a part of, really every biological process on Earth.
So what would the world look like without microbes? Can you even imagine it? Would we even be able to exist without them?
Yeah, that is a really good sort of thought experiment when you start to think about just generally biodiversity. One way I tell my classes to think about it is imagine what types of organisms you'd have to bring with you if you were going to set up a colony on the moon, for instance. Of course, you could first start to think about, well, I need food and the plants, that I might eat, and maybe the pollinators the insects that are pollinating the plants. But then when you start to think about, well, we need to have soil up there. Well, there's lots of microbes that are in the soil recycling nutrients. We want the microbes there to help decompose our waste once we're on the moon and purify our drinking water and all of those microbes. So we don't actually have a good understanding of if we had to pick out the different microbes we would want, or diversity, if we had to reestablish a community, an ecosystem by scratch.
When we're talking about microbial community, what usually is in this community? What types of organisms, and is there different species within a community, or is it just one species, and do they mingle or interact?
Mm-hmm. Yes, so it depends on what type of environment you're in. If we just take the soil, there's really there are billions of cells in a handful of soil, and we think there's thousands, maybe even up to 1 million different types of species of bacteria in that soil as well. So there really is an incredible variation of life there that we can't see. That changes depending where you go. So if you go to the ocean, that diversity is less. It's still very high, but it's less. And so we have a better understanding of who's there at this point. And then if you look in your gut, for instance, in your own microbiome in your gut is much lower. It's now estimates are in the thousands of bacteria that are there. So I was just giving numbers for bacteria, but there's also fungi, there's viruses, as I mentioned, there's this group of organisms called archaea that are related to eukaryotes and other single-celled organisms. So there's quite a few different types of microbes.
So the microbial community or diversity is very important for the environment and the environmental processes. And so how are they responding to changes that happens in the environment, or how are those affecting the microbes?
Yes, so we can see that the microbial community, the composition of that microbial community is changing as you move from environment to environment. So the microbial community in an agricultural field in that soil is going to be different from the microbial community that you see in a desert soil. So we know they're responding to the environment. The next question is, can we predict how they might respond if the environment changes? If we go through another drought year, for instance, in California, how does the composition of the desert soil or the forest soil, how does that change? And then the final question is, okay, if it's changing, then does that make a difference for many of the processes we care about that those soils and plants are carrying out in the environment?
When you're thinking of microbes, is it a very different way of thinking to studying other organisms like plants? Like, how do you get into the tiny mindset, the tiny world mindset?
Yeah, I think that's a challenge. I mean, I personally am always trying to think about a microbial community and picture it like, what would this mean for trees, or something that I can conceptualize and see. And I try to explain it to people that way too. But you're exactly right. It's so abstract that sometimes we can forget these basic things about, what it must be like to be a really small cell and the sort of physical forces that cell is experiencing. It's easy to forget about that because it's very different at that level. And I'm not a physicist, but there are other people that study exactly that. Thinking about how life is really different at a small size.
Yeah, because they're not like just tiny us.
Yeah, I was talking to Lily before. It reminded me of Dr. Seuss' Horton Hears a Who.
Uh-huh.
And I don't know why, when you were talking about microbes, I just thought about that movie and how there's a world in that little speck.
Yeah.
So to me, it was like that.
Yeah. And mostly we're taking so many of them, like literally billions, and grinding them up and then saying we're trying to understand them. And maybe we're just looking at the wrong scale to get the answer, because they might be controlled by things that we're not even, able to measure the differences across soil particles or something like that.
So your lab focuses on the diversity of microbes, and they're so tiny. Like, how do you study diversity, and how do you quantify, like, if something is very diverse versus something that's not as diverse? How can you tell?
Yeah. Well, that's really why we, at this point, still know so little, is because we haven't had the techniques to tell them apart. So if you look at soil bacteria under a microscope, most of them look exactly the same, small and maybe rod shaped. Maybe there's a couple that are more circular. So it's pretty hard. You're not going to be able to use their morphology to distinguish them. And so the huge breakthrough that happened a couple of decades ago is that we're able to apply some molecular genetic techniques to distinguish them apart. So we essentially are sequencing their DNA, and from that we can classify them into different groups. And that was the big breakthrough to study diversity. Yeah.
So when you are counting them, does it matter if they are dead or alive? I was just curious on how that works.
That's a great question. Yes, it should matter a lot. And so there are people that are trying to figure out good methods to distinguish between microbes that are dead and alive. Because, of course, if we're using DNA and the genetics to characterize them, that DNA could be staying around for a while, especially certain environments. And so we may not be characterizing who's really active there in a community. And we might be getting, again, this a false picture of what's going on, at least that we at this time right now.
Can I ask kind of a spooky question? So,
Sure.
what does death look like in a tiny little bacteria or a tiny little fungi? Do they just stop functioning and leave behind a little microcorpse?
Well, I'll answer for bacteria because that's what I know more about. But yeah, it would mean that they stop dividing, right? So it is another area of research of how long can a cell last without dividing, but just sort of be dormant, right? And hang out. And it's probably a lot longer, especially in something like soil, than we might usually assume. And so really, yeah, you have to wait until you're really sure that the cell is not dividing anymore. And that might be obvious if the cell membranes are breaking down, right? And the guts are spilling out, essentially. But other times it may not be, if the cell looks intact.
When you're looking at microbial diversity, how do you set up an experiment to study microbial diversity and how it's different in different areas?
Yeah. Well, as I mentioned, a lot of what we're doing is just natural history and characterizing what diversity is out there. But beyond that, we might want to experimentally test some hypotheses, and that might be whether or not one community works differently than another community. And so one of the main ways we've been trying to do that is to use what I call our microbial cages in order to move communities around and kind of trap them in a cage so that we can place them in different environments and see how they work, how they function. And so our cages, of course, the sort of mesh for the cages is very, very small. It's less than the openings of the mesh are less than the size of a bacterial cell, so that the bacteria we put in there don't get out and other bacteria don't get in. So that's how we've been trying to test how different communities work right now.
What Dr. Martiny did here with this cage experiment is to test whether microbial communities function the same way in their home environments as in other environments. To do this, she puts the community in a mesh cage.
The mesh cage not only enables her to move the community to a different environment, it also keeps the native microbes of that environment out. In this way, she is able to test how the community responds to a new environment.
So, what are some of the experiments that you have going on in your lab, and what have you found so far?
Yeah, so we had a really fun experiment recently where we wanted to test whether different microbial communities from the surface of soil work differently. And that's a really hard question to ask in the environment because microbial communities vary very closely with their environment. So if you just compare what a microbial community looks like and how it's working, there's this covariable all the time with its environment. So somehow you have to tease apart the environment and the community. And that's where it comes, where I mentioned these microbial cages. That's been our approach to try to do that. So we can inoculate a community inside this cage, and we can move then the cage around to different environments. So we use that approach across an elevation gradient in Southern California, moving from a desert near Palm Springs all the way up to an alpine, subalpine area with pine trees. And in this way, we were able to tease apart how much decomposition, so the degradation and of leaf litter, dead leaves right on the surface, whether that changed depending on which community we put in it, and also how much that changed across the environmental gradient. So those are the types of experiments that we're doing at the community level these days.
And so what are some results? What have you seen so far? How are these communities changing in response to the different environments that they're growing in?
Yeah, so, well, we see, as we would expect, that the environment itself, by itself, is really important. We had no doubt that that was the case. And so the measure we were using is decomposition of dead leaves. And we know that if you add more moisture, for instance, and the environment is warmer, in general, decomposition happens faster. So we saw that effect. But when we also controlled for the environment, kept the moisture the same, kept the temperature the same, we saw that different communities also had different rates of decomposition. And the interesting thing was it was sort of unexpected. We still can't explain why some communities in some environments seem to do much better. They decompose this leaf litter faster than others. And so that, of course, we've answered one question, that the community composition matters, but it's opened up all these other questions about why it matters. And so now we're trying to dig into, what is different about this community, and what are the features of the microbes that are causing decomposition to change.
And so your studies involve moving communities from different areas into new areas that they don't usually experience. And so my question kind of is, so you did, you had a community from the desert and then subalpine communities and putting them in different environments. Does this give you an idea of how microbial diversity is going to change in response to climate change and warmer climates and drought?
Yeah, you could use these results to ask, how adaptable in a way the communities are. And in fact, when we move the communities across this whole gradient, it's not as if they're in shock and die off. They do still function pretty well. It takes them a year or two to start to look like the surrounding community. And so that's sort of an interesting result. And so, yeah, we can use some of our data to try to make predictions about, as temperatures get warmer and precipitation falls, at least it gets less in Southern California, what do we think these ecosystems are going to look like and how are they going to work?
So it's somewhat just similar to, say, we move to a different area with a different climate and we need that adaptation period. It seems like that's going on with the microbes as well.
Yeah, there's a few different ways that you can adapt. You are talking about, well, I'm going to move to Machu Picchu and I'm physiologically need to acclimate to the high elevation. And there's some of that in microbes too. There's also that the community as a whole can adapt in that it shifts the population size of different members of the community. And then finally, you might even see evolutionary adaptation because there's so many of these organisms and their generation times are relatively fast compared to humans. They can adapt pretty fast too. So it is an interesting question to think about how all these processes are going to work together under some future change in climate. In trying to predict what's going to happen.
And so you said it takes about 2 years for the community in your cages to look like the surrounding community. But what does that mean? Are they gaining new members, or are they functional? Yeah, are they interacting somehow, or is it their function is starting to look more like the surrounding community?
Yeah. Well, we try with the cages to prevent the intermingling, but it is possible that we are not, that small holes start to open up in the cages and microbes come in. So that's one possibility. However, what we think the main effect going on is that the communities are changing in their composition because some microbes really like the new conditions, so they grow better than others, and others really hate it and they start to die off. So you see big population shifts of who's there and how well they're doing, and that's how they tend to merge towards the new communities there.
So does this mean that in all of the different areas, they're composed of the same like types of bacteria and fungi, just in different abundances?
Well, that's a great question. Also, I think in our gradient that's probably the case. But the problem is it's really hard to say for sure that something's not there, right? So there's so many cells, just even in our little cages, that I can never tell you for sure because I can't sample them all to say that, this subalpine organism is not present in the desert. But what we've seen so far is that kind of, generally speaking, everything's kind of there. But this is a rough, really, even though our techniques in a way are sophisticated by doing the sequencing, it's still a very rough assessment of the community. So I can't tell you for sure that whether an organism is there or not, and I can't, it may be that it looks pretty similar, but actually was a different strain than the other one, and so we're not really able to say that. We'll need some different experiments to test that out.
And how are you able to know whether the effects of a change are because of the community or because of something that happened in the environment?
Yeah, so we have to vary both things in all directions. So we have replicates of the same community many times in one environment, and we compare that to replicates with another community in the same environment. So we're either controlling for the environment or we control for the communities, and in this way we can tease apart both effects.
Dr. Martiny is talking about her experimental design, the way that she sets up her experiments to answer her questions about microbial diversity. For an experiment to be sound, there need to be replicates and controls.
Controls are groups that don't have an experimental treatment. That is, they're just existing as they are. For Dr. Martiny's experiment, the controls are the groups of microbes that are placed in their home environments, and the experimental groups are the ones that are in different environments. You compare the experimental groups to the control groups to see if there was a change.
Replicates mean you have more than one group receiving the same treatment. That way, if all the groups receiving the treatment are different in the same way from all the controls, it is more likely that the treatment is causing that change.
So these are like preliminary studies, and you have a lot more work to do, but can I ask, what do you think this means for the environment?
Well, these microbial communities are different in these environments already. These experiments are really to try to understand on our side, can we predict how fast decomposition or any other ecosystem process is happening? Right now, our models of ecosystems, they're so-so. We get it right some of the time, but a lot of times we don't. And so I guess a lot of our research is trying to see whether or not maybe if adding microbes into the equation can help us understand those processes better. And in fact, a lot of the processes that are going on in ecosystems are driven by microbes. Many times, nitrogen cycling, how nitrogen is transforming between the atmosphere into the soil, or decomposition, like I mentioned, that's really moving carbon between the atmosphere and soil as well. All these types of nutrient cycles, microbes are playing a huge role in those. And so if we're only studying plants and maybe some just general features of temperature and rainfall, we may be missing a lot of the picture because the organisms that are kind of the arrows transforming these nutrients into different forms, if we don't know anything about them, maybe knowing more about them will help us solve the puzzle and make our predictions better in terms of how fast these rates are.
What are some common misconceptions you hear from the community or from society about microbes, and what do you wish you could tell them about how important they are?
Yeah. Well, the first one is that not all microbes are germs, right? That's typically what people think about, is that they're bad. And that's how they get sick, right? When you talk about bacteria and viruses. But the vast, vast majority of different types of microbes, and in terms of numbers, in terms of diversity, they are not harmful at all to us. And in fact, they're really essential for lots of processes that go on in the world. Everything from, as I mentioned before, waste decomposition, water purification, but even, our own digestion, that's essentially just decomposition inside of ourselves. These are important, either symbiotic or commensal, meaning it doesn't harm us. And it does, and it may not help us either, but they're just traveling along with us. So that's most of microbes out there. And so at least when I'm talking to community audiences, that's the point I tried to drive home, that this is a huge area of biology that a lot of people don't know about, that is really essential to how the world works.
Yeah, I saw in one of the images on your website, it has the words, "If you don't like microbes, you're on the wrong planet." So I thought that was an interesting
Yeah, they're pretty unavoidable.
So. Do you have, or is there any sort of fun facts about microbes in our daily lives that you would want to share with us? Or something you might tell someone and they're like, "Oh, I didn't know that."
That's really cool.
Yeah. Like, that's really cool.
Fun facts about microbes. There's so many. How can I choose? Well, one area that we haven't talked about at all really are viruses, and I really like viruses. I especially like viruses that infect other microbes, especially bacteria. So those are called bacteriophage. And in my work on that, we studied them in the ocean. So in the ocean, there are 1 billion viruses in a liter of seawater. So that's something to think about next time you go swimming in the ocean. And again, remember, they're not germs. They don't infect you. They infect other microbes in the ocean. And so they're really an important part of our ecosystem there. But yeah, that's my fun fact.
So you think you're swimming in salt water, but it's really virus water.
A sea of microbes and viruses, and they're happy. They don't care about you. Yeah.
Awesome.
Well,
thank you.
Yeah, I think that's all the time we have. Thank you so much for joining us today, Dr. Martiny.
Thank you for having me. It was great.
Thank you.
This episode of Radiobio was produced by Sonia Vargas. The interviewers were Lily Pennington and Saninta Pertiwi, and the episode was edited by Saninta Pertiwi and Sonia Vargas. The artwork was created by Ann Diep.
Radiobio is produced by graduate students at the University of California, Merced. Support for Radiobio comes from the Quantitative and Systems Biology Graduate Group, the School of Natural Sciences, and the Graduate Division at UC Merced. You can help support Radiobio's mission of increasing scientific literacy in California's Central Valley and beyond by donating at giving.ucmerced.edu/radiobio. Find out more about our mission, events, and podcasts at www.radiobio.net.